Let us wait for other students to join and then we can begin from 5 minutes from now on. Hello, let us wait for 2-3 more minutes for the others to join.
Yeah.
Can you please mute yourself? You can switch on your mic when you have anything to say, okay? When you have any doubts, please switch it on.
It is. Each of. Hello, ma'am.
Hello.
Are you listening? Ah. One doubt is here. How to find out PKB when Ka is given for weak acid? So how to calculate this? If
Yeah, I will teach all of that in this course. Okay, so I will
uh,
tell you a little bit of the theory and then we will do question answer. That time you
uh,
can ask me.
okay.
Okay. Okay. I am muting you for now. You can unmute yourself whenever you have a doubt. Okay. Okay, so now it's almost 6 9. So let us begin. Hello all, I am Beas Mukherjee. I am a research scholar from IIT Kharagpur and today we will be discussing the question and answers of week 1 session of the NPTEL course Experimental Biochemistry, which is taken by Professor Shagota Dasgupta and Professor Shomode of IIT Kharagpur. So in this course, I will be discussing the theory portion with you and then we will be solving the questions that are related to that portion. In this way, it will be easier for you to catch up. And since this is an interactive session, you can type in the text box, you can answer in the text box and also you can switch on your mic and you can talk. Okay, you can ask me questions. So let us begin. So for this course, I will start with very selective questions of the week 0 assignment. I am sure you guys have already got this assignment, you have solved it and you have probably even got the solution for this assignment. So I have chosen only a few questions that are very relevant to the topics that have been taught to you in this week. So we will solve those questions and then in the week 1 assignment, we will solve all the questions together. Okay, so first is the first question. So whenever you get into a lab, a biochemistry lab, the first thing that we do early in the morning is we clean our workbench. Okay, and so the first question is what percentage of alcohol should be used for surface sterilization? So the options given are 100%, 90%, 70% and 50%. You can give your answer in the chat box. Please give your answer in the chat box.
It's 70%.
70%. Okay. So yes, you're very correct. Let us find out why that is so. So sterilization is actually a process in which you eliminate all forms of microbial life. Alright, and that can be done by physical and chemical means. So why exactly 70%, why not 90 or 100%? Let's see. That is because in case of 70% alcohol, since water is present, this alcohol is actually able to get inside the cell. And what it does is it denatures the enzymatic structure of the different proteins present in the bacteria. As a result, what happens? The bacteria is dead. Okay. But then in case of 99%, that is absolute alcohol, what happens? It is so dehydrating that it cannot enter into the bacterial cell. Instead, it just burns up the bacteria. But then it renders it dormant. It does not completely kill it. And therefore 70% of ethanol is more effective. And that is why these hand sanitizers that we used to use during the COVID-19 breakout, all of these actually contain 60 to 70% alcohol. And those were in fact very efficient in killing these envelope viruses like coronavirus. So let us come to the sterilization slide. Now there are the different methods of sterilization. If you are interested in them, you can actually look up. It is given in details. See here, the physical methods are there and the chemical methods are also there. So in the physical methods, you find heat, radiation, filtration, etc. I'll choose a pointer from here. Okay, so that it becomes easier for you to understand which portion I'm referring to. So all of this you can look into, and alcohol that we were talking about, this comes under the chemical methods of sterilization. Alright, now we move on to the next question. Now these are some very basic calculations that we perform every day in a biochemistry laboratory. Now here what the question is, we are seeing that you've been asked to make this 70% ethyl alcohol volume by volume. So volume by volume means 70, if in a 100 ml solution, 70 ml has to be ethyl alcohol. Okay, and that you have been asked to make for what? 280 ml. So how much alcohol, how much absolute alcohol will you need for that? So try to solve this question. You can chat, you can give it in the chat box, or you can even switch on your mic and you can tell me the answer. So I can give you a hint. See, for 100 ml, 100 ml of total solution, if you need 70 ml of ethanol, then for 280 ml of total solution, how much ethanol would you need? That is the solution. Okay. So in the next question, let us see. I will come to the solution. It will get displayed very soon. So the next question you can try. This is again a very simple question where you've been asked to make 0.6 molar NaOH solution, and that also of volume 250 ml. So for that you have to calculate exactly how much NaOH you need to weigh, because NaOH comes in the form of a powder. So to make this 0.6 molar solution of 350 ml volume, how much NaOH exactly do you need to weigh? So for that you need to recall the formula for molarity and you need to find it out. So you have 2 minutes to solve these two questions. Hello, we are solving questions here. You can answer in the chat box, or you can switch on your mic and you can tell me the answer, alright? So these are the two questions that we are looking into right now. In the first question, they have asked you how much of ethyl alcohol in volumes would be needed to make a 70% solution of total volume 280 ml. Okay, in the second one, they have asked you to make a 0.6 molar NaOH solution of 350 ml volume, and for that how much of the NaOH powder you are going to measure. Let us look at the answers. So the answer for this one is 196 ml. And why is that? For this you can use the C1V1 equals to C2V2 formula, in which C1 is your absolute alcohol, right? So that is 99%. That is C1. So here C1 becomes 99% and V1 is how much volume you will actually take from this to get this V2. V2 it's given, it's 280 ml and C2 is 70%. So that is 99 into V1 is equal to 70 into 280. Right? So if you do the calculation, you get 196 ml. So in the next question, what we do is, we use the molarity formula to find out exactly how much of NaOH powder needs to be measured. Now see, for the molecular weight of NaOH, it is 40, right? So it is given as 40 gram per mole, and to make a 0.6 molar solution, you need exactly 8.4 grams in 350 ml, so that you can get this 0.6 molar NaOH solution. All right. So in the next question, we will be talking about pH. So I will explain to you pH in more details very soon. We just have two questions here which are very basic, and these can give you a brief idea about what pH is. So this is a very common question. What do you think is the pH of pure water? You have 1 minute to answer this. You can type in the chat box, or you can tell me by switching on your mic.
Uh, pH of water is 7.
Yes, very good. Yes. So why exactly is it 7? Why is it 7? We'll soon find out, okay?
Uh, Here is the neutralized
Yes.
is neither acid or neither base.
Mm, we have a different explanation. Very soon we are going to find out. In assignment 1, you will actually come to know exactly why it is 7. Alright. So,
Yes, yes.
hmm. Okay, so now this is the next question. What is the pH of 0.6 molar HCl? Now if you look into this question, HCl is actually what? It is a strong acid. So a strong acid, whenever you put it into water, what happens? It completely dissociates, right? It dissociates into H plus and Cl minus. So there is no equilibrium state. It completely gets dissociated. Therefore, what happens? Whatever initial molarity was there in HCl, it dissociates into H plus and Cl minus, right? So that will also have the same concentration. So now find out what will be the pH of this 0.6 molar HCl. We will look into details very soon. This is just to give you a very brief idea. Okay, so I'll tell you the answer. You're very right. The correct answer is 7 for this question. And for this question, the correct answer is 0.22. Let us see why. So yeah, HCl being a strong acid, it completely dissociates into H plus and Cl minus. So the concentration of H plus. So what exactly is pH? pH is nothing but the negative logarithm of H plus ion concentration. So if I tell you that initially the concentration was this 0.6, and then when it dissociates, it is again 0.6 and 0.6. So 0.6 moles of H plus and 0.6 moles of Cl minus. Then we take minus log of 0.6 and we calculate that and we get 0.22. Right. So this HCl reacts with water. Water here is acting as a base. And then since this is an acid, the basic definition of an acid according to Bronsted and Lowry concept is that it releases an H plus ion. Right. So this H plus ion reacts with water, which is the base, and it forms the hydronium ion. Now we need to find the concentration of this H plus ion to determine the pH. and this Cl minus that gets separated, this forms the conjugate base, which is the acid base pair, right? So this is all. Now we go to the week 1 assignment. Here we will be dealing with all the different concepts and we will be solving the questions accordingly. Okay. So firstly, what is accuracy and what is precision? Now let's say that you and your friends go to a fair and they have asked you to play this game of darts. Okay, they have asked you to hit exactly this circular yellow zone. Now let's say you are very good with the game of darts and you hit exactly in the yellow region. All the 4 darts are in the yellow region. Now they say that you are both accurate and precise. So next your another friend goes and hits, but then he is not very good with the game and he hits very close to this yellow region. So we can say that he is accurate, but then he is not precise because see the places that he have hit, this is quite far away from one another. Again, there is one more very unlucky friend who is really good with this game of darts, but then he has not understood where exactly he is supposed to hit. So he has hit some in the blue region, and but then all the darts are exactly at one place. So then he is not accurate, but then he is precise. And another friend, he is just there for the fun of it. He has hit wherever he has want, and it is all over the board. So this friend is neither accurate nor precise. So from here we can understand that accuracy is how close your measured value is to a true or known value. Okay, that is accuracy. And precision is whether if you're trying for, let's say you're trying it for 4 times or 6 times, whether all of these 4 measurements or 6 measurements are very close to one another. If they're very close, then you are very precise. Otherwise, you're not. Okay. So these two concepts are very important in biological experiments, because in biology we need to confirm that the experiment is correct through reproducibility and repeatability. Okay, so if they are reproducible and repeatable and we get similar results, then we can confirm about the accuracy as well as precision if the values are consistently similar. Okay, so now here you can get a more detailed idea on accuracy and precision. Now comes in error. So what exactly is error? Error is the difference between your measured value and the true value or the actual value. Okay, so now there is a question on accuracy and precision. Let's see how accurate you are with this question. So this question says that 2 researchers have been asked to measure the pH of a buffer. Okay, and the buffer that is given to them is 7.4. Now researcher A measures all of these values and researcher B measures the values and says that these are the different values that are there of the buffer. Okay, so who do you think is more accurate and who do you think is more precise? Now, if you look into this question, researcher A, the difference between the values that he has measured, this is quite a lot. But then in case of researcher B, the differences are very minute. It is just plus minus 1 from the true value, right? So the true value is this and he has got that twice. So that means he is very close to the true value. That makes him quite accurate. So the standard deviation is very less in this case. Alright, and that tells us that also precision is pretty high. Here the standard deviation is very high. See, the values are very different from one another. And also none of the values are very close to this 7.40. So let us see what the answer says. So the answer is A is less accurate and precise than B. That is, B is both accurate as well as more precise compared to A. Why is that so? Okay, so firstly they have calculated the average for researcher A and researcher B's values, and then they have also calculated the standard deviation for both. Now if you see, this average value is close to 7.40, but it is not exactly 7.40, and the standard deviation is also quite a lot compared to this value, which is the standard deviation for researcher researcher B. For researcher B, the accuracy is very high because the average of the values that he has received, it is exactly 7.40, which is equal to the true value, right? So the average of B is closer, and so B is more accurate. Similarly, the standard deviation for B is less, and therefore B is more precise. Now we go to another concept, which is pH and its significance in biology. So now you look at this scale. pH is a scale to measure the acidity and basicity of a solution. Okay, so it is a scale from 1 to 14. So as you go lower on this scale, the solutions are more acidic. More acidic meaning they are more sour. Okay, sour in taste. And then as you go from this neutral value towards the higher values of pH, the solutions are more basic. So what happens is the solutions that are more acidic, they are having more concentration of H plus ion. That is, pH is after all just the measurement of the H plus ion concentration in a solution. Okay, so the pH around this region, around 7, is actually neutral, and at room temperature pure water has a pH of 7. We will find this out very soon. Why exactly it is 7, we will find out in details. Alright, so the solutions which are very close by to this neutral pH, they are less acidic and less basic. But then as you go farther from the neutral pH, the solutions become more and more acidic or more and more basic. Alright, so pH is calculated as the negative logarithm of H plus ion concentration, which can also be written as the logarithm of 1 by H plus ion concentration. Next, let us see. So if you remember your school time experiments, we had measured pH of solutions using this small paper, which used to change color based on the pH of the solution into which it was dipped. Now this was called litmus paper. So there is blue litmus and red litmus paper, right? So when you dip blue litmus in acidic solution, it turns red, and when you dip red litmus in acidic solution, this becomes blue. But then the problem with litmus paper is that it is not temperature sensitive, but then pH of a solution changes with change in temperature. How exactly is that so? Let us find out. So pH of a weak acid decreases as the temperature increases. Why? Because as temperature increases, the vibrational motion of the molecules also increases. As a result, what happens? More dissociation takes place, and therefore the H plus ion concentration increases. If H plus ion concentration increases, the solution becomes more acidic. As a result, what happens? And pH is the negative logarithm of the H plus ion concentration. As a result, pH decreases, the solution becomes more acidic. If the solution is a weak acid, then the temperature rises. Now pH plays a very important role in our body, because our blood is having a pH of 7.4, and it is a buffer system where pH is very tightly regulated, and the cells and then the cytoplasm is having a different pH, the extracellular matrix is having a different pH. So pH very tightly regulates the different cellular functioning in our body. So the cytoplasm of the cell has a pH of around 6.8. Now you can refer to all of these examples and find out the pH of different forms of things. So if you look at this, seawater is not exactly having a pH of 7. Rather, it is having a pH of 8 because of the different salts dissolved in it. Okay, so all of this is very interesting. And then the gastric juice, that is HCl in our stomach, this is having a pH of nearly 2. So that is very acidic. Right. So pH meter. Now let us try to answer a question about pH meter. So pH meter is a device that can measure the pH of a solution very accurately, and it also takes into consideration the changes in pH due to difference in temperature. So it is lot more advanced compared to litmus paper. Alright. Now they have asked you, in case of a digital pH meter, which of the following paths can you see? Now, if I tell you about a pH meter, what it does is, there is actually a voltmeter which is connected to the two electrodes. One of them is a reference electrode and one of them is a glass electrode. Now, this voltmeter actually measures the hydrogen ion activity or H plus ion concentration, and then it translates that information into pH information, and then it gives you so it actually measures difference in voltage measured by these two electrodes, and then it converts that information into pH information and gives you the pH of the solution. Now, looking at this figure, what do you think is the constituent of a digital pH meter? Which one do you think is the right answer? First answer says there is a glass electrode and there is a monochromator. We do not see any monochromator here, right? It is there in what? Spectroscopic measurement instruments. So it is
Glass
not
electrode.
there. Which one do you think?
Glass electrode.
Glass electrode, yes. And
Mm, temperature sensor.
very good. Yes. Glass electrode and temperature sensor. Right. So now we go to one very quick question. This is, find out the concentration of H plus ion in 1 millimolar NaOH solution. So 1 millimolar means it is 1 by 1000 of a molar, right? So in 1 millimolar NaOH solution, what is the concentration of H plus ion? So let us, you tell me.
Madam, actually, I don't know the formulas for that.
Okay, no problem. I will explain. It's okay. So for this, let us find out how exactly we can calculate this. So pH and pOH together gives you 14. Okay. So the right answer is 10 to the power 11, 10 to the power minus 11 molar. Why exactly is that so? Because for 1 millimolar NaOH, the concentration of OH minus ion, this is actually 10 to the power minus 3 molar, because it is 1 millimolar, right? So that is 10 to the power minus 3 molar, and that is true for OH minus ion concentration. So now if we know that OH minus ion concentration and H plus ion concentration together makes up this 10 to the power minus 14, which is true for a solution at 25 degrees centigrade, and that is why what we see here is H plus ion concentration comes to be 10 to the power minus 14 divided by 10 to the power minus 3, and that is 1 into 10 to the power minus 11 molar. So right answer is 10 to the power minus 11 molar. Now we will look into another concept, which is how is the acidity of a solution quantified. Now this is what you were actually asking me about, this Ka and pKa. So we will look into this in details. Now I give you a break of 5 minutes and then we will come back and discuss this, all right? So let us take a break for 5 minutes. Hello, so we are back after 5 minutes of break. Let us begin with the questions. Let us carry on. So, we were looking into this, how is acidity of a solution quantified? So, to understand this, the acid dissociation constant. So, okay. So, here if you look at this equation, HA is the acid, where H is the H plus ion that is bound, that is connected with this conjugate base, which is the A, which is the molecular group. Okay, and this molecular group releases this H plus ion and it forms this A minus, which is the conjugate base of its acid. Now what happens, this is, you can see that this is a reversible reaction, so there is both forward and backward reaction. So at equilibrium, that is the K equilibrium is equal to the concentration of H plus ion, concentration of A minus ion divided by concentration of HA ion. So at the equilibrium, what happens is both the forward reaction and the backward reaction are happening, but then they are happening at the same rate, and that is why what happens, the concentration of H plus and A minus is equal to the concentration of HA. Now this is termed as the equilibrium constant or the acid dissociation constant. Now we can take the negative logarithm of this acid dissociation constant, and from there we can find out the pKa of an acid. So this pKa is actually the quantity that is used to determine, it is the parameter that excuse me, this parameter tells us how strong an acid is or how weak it is. So if Ka is very high, that is the dissociation constant is very high, that means more of H plus ion and please excuse my throat condition. Yes. Okay. So we are carrying on with this. Right. So when Ka is very high, that is the dissociation constant is very high, that means more of this acid has been dissociated into H plus and A minus already. And that tells us that Ka is very high, pKa, which is the negative log of Ka, it is very low, and therefore the acid has largely dissociated and therefore it is very, very strong. Okay, so strong acids have a low pKa and very high Ka value. Okay, and when the Ka is very low, so that means what happens, more of this acid is remaining and less of it has been dissociated into H plus and A minus ions. So that means for very weak acids, what happens, the Ka is very low and therefore the pKa value is very, very high. So weak acids have a pKa from -2 to around 7. So now comes in Henderson-Hasselbalch equation, which is one of the most important concepts, and Henderson-Hasselbalch equation actually relates pH to this acid dissociation constant or Ka, and the pKa also as well with the concentration of the A minus and the acid. So this is the equation which tells you that pH is equal to pKa plus log of this conjugate base by its acid. So now we will look at this question. So why exactly? So we already have solved this question. Gopinath has solved this question for us, right? So this is a question on pH of water. So pure water is neutral, but why exactly is it so? So the first option says it has pH 7 in experimental conditions. Next says that it has equal amount of H plus and OH minus ions. Third one says water does not contain free H plus and OH minus ions. Fourth one says water cannot ionize. So to look into this question in deeper details, let us see. So at 25 degrees centigrade, the concentration of water has been calculated to be 55.5 molar. How are we doing this? Because we already know the molecular weight of water. Molecular weight of water is 18 because there are 2 hydrogen and 1 oxygen. So for each hydrogen it is 1 and for oxygen it is 16 and therefore it is around 18 grams per mole. Alright, so that is the molecular weight of water, and then from that value if you calculate, so 100 gram has been taken, 100 gram per liter, and from there we get the concentration of water. Now we know that water actually dissociates into H plus and OH minus, although this is actually hydronium, but still let us consider this as H plus initially. So water dissociates into H plus and OH minus, and at equilibrium concentration, if you substitute this concentration of water with this value 55.5 molar, then what we get is this Kw, which is the ionic product of water. So ionic product of water is nothing but this value K equilibrium multiplied with this value. So this product H plus and OH minus from water, right? This product is termed as the ionic product of water. Now, actually, this ionic product of water, both the values, this is also, this H plus concentration is actually equal to H minus concentration. See, this is the answer. So, what difference does it make? So, let us find out. So, in this ionic product of water, if you see, if these two values are equal, then what happens? It becomes H plus concentration whole square, right? So, if we solve the actual H plus concentration from here, then we will get, so we know that the final uh, pH for water is 10 to the power minus 14 moles, that is the final concentration, and then if we take the square root of that, we get the concentration of H plus and concentration of OH minus is equal to 10 to the power minus 7 moles. So then 10 to the power minus 7, from there if we can find out the pH, that is negative log of 10 to the power minus 7, we get this value, which is, see, pH is equal to log of this, and then we get the value log 1 plus 10 to the power 7 log of 10 to the power 7, and that gives us 0 plus 7, right? So that tells us that, and the pH is 7 for water at 25 degrees centigrade. So now in the next question we will be looking into Ka and pK. So did
Excuse me.
that solve your question?
Hello, madam.
Yeah, did you understand whichever question you were asking? Did that
So
clear
I have
or not?
show the screen of your computer, please. So then I can be taken. It is that.
Yes.
Uh, so I can see the what is going on there.
Huh. Can you see the screen?
I'm not seeing the screen of your computer. I'm only seeing
One
your.
minute. Oh, okay. 1 minute. Can you see the screen now?
Not it. Not it.
1 minute.
Ah, yes.
Can you see?
Now right. It's the most important. Sorry for that.
Hmm. Right. 1 minute. Yeah. So yes. Yes. Can you see the screen now? Yes. Can you tell us the question again? What is your doubt?
how to find out the K PKB where K is given.
Okay, so how to find out PKB when Ka is given, right? Okay, so that is the doubt. All right. So this is the question.
Uh-huh.
That Ka is actually given to you, and Ka of the weak acid is given as this, and then they have asked you to find PKB. Now, from Henderson-Hasselbalch's equation, you know that, 1 minute, yes. So, Henderson's-Hasselbalch equation, you know that pH is equal to pKa plus log of conjugate base by acid. Now, the same thing, now this is for your weak acid, whereas the same thing can be calculated for weak base, for which this POH will be equal to pKb plus log of conjugate acid divided by base. Okay, so that will be the equation for pKb. And also there is one more thing. Now this pKb will be done for weak base. And another thing is pH and POH together gives you 14. Okay, because in pure water, this H plus ion concentration and OH minus ion concentration, you know that it is 7 and 7, so that total gives you 14. 10 to the power minus 7 and 10 to the power minus 7, and that is why the ionic product is 10 to the power minus 14. So for using all that, keeping all that in mind, we will try to solve this question. So they have given you the Ka and they have asked you to find the pKa. So since they have given you the Ka of the weak acid, you can very easily find out pKa from here. So how will you do that? You will take a negative log of this value. Alright. So you get pKa from there. And so from there, you can actually calculate the pKb. So here let's see what the solution says. Yes, so pKa is minus log of Ka. So pKa has been given to you, Ka value has been given. So pK you find out as 5.25, and then pKa plus pKb together gives you 14, and therefore pKb is after all 14 minus this 5.25. So the value is 8.75. Okay. Did that solve your doubt? Yes, so the connection was lost and we are back again. Let us look into it. Okay, so yeah, so this is how we solved it, and then we will look into the nature of different acids. So according to the Bronsted-Lowry concept, an acid is a molecule that is capable of donating a proton. Okay, so the molecule that is capable of donating only one proton is termed as a monoprotic acid. Now, if the molecule is capable of donating two protons, it is termed as a diprotic acid. If the molecule is capable of donating more than one proton, it is known as a polyprotic acid. So, now if you look into this graph, this tells you that when a weak polyprotic acid is titrated, and we see that initially, okay, so we see that there are more than one pKa values. That is because more than one protons are being donated, and then these are the equivalence points. So in the equivalence points, what happens is the concentration of the conjugate base and the acid, these are similar. And so at the first equivalent point, the concentration of the conjugate base and the acid has reached in such a point that sufficient base has been added to neutralize the first acidic proton, and it gets released. In the second one, you see this is the cell buffering range. That means that around like a zwitterionic substance is present, and so it is very difficult to change its pH even on addition of charged entities. Now again, in the second equivalence point, the amount of base that has been added, it is sufficient to neutralize the second or the third proton, and therefore it is released. So this is how polyprotic acids work. Now this is an MSQ, that is multiple select question on Ka and pKa. So there are 4 sets of solutions of different pKa values. So which of the following statements is or are true? So the name of the compounds are given A, B, C, D, and these are the pKa values of the 4 compounds. Alright. So which of the following statement is or are true? So acid A dissociates to a greater extent in water than B. Now C and D are basic in nature, whereas A and B are acidic in nature. D is the conjugate base of C, and A is more likely to be a polyprotic acid compared to B. Now if you look at A, it has only one pKa value. Now we are not sure because they have not given more than one pKa value, so we cannot say whether it can really be a polyprotic acid. Now the next one, A dissociates to a greater extent in water than acid B. So that is possibly true because the pKa of this is quite low. So that tells us that this is a stronger acid. So if pKa is low, that means Ka is high, and if Ka is high, that means at the same pH, this particular acid definitely dissociates and gives more H plus ion compared to this acid B. Alright, and looking at the pKa values, we can say that both A and B are acids and C and D are alkaline in nature, right? So C and D are basic in nature, whereas A and B are acidic in nature. So that tells us that definitely these two can be correct. D is the conjugate base of C. No, that is not possible, because conjugate base will be more basic compared to the acid, right? So that cannot be true. Both of these are basic in nature. So let us find out. The answer is both A and B. Alright. So we move on to the concept of buffers. So a buffer is a solution which is very resistant to pH change. So what happens in case of buffers? Even when we are adding OH minus, let's look into this acidic buffer, which is acetic acid. Okay, so when we are adding OH minus, it actually combines with the H plus ion that the acetic acid releases, and then it forms H2O. And this conjugate base is remaining. So what happens if more of H plus gets consumed in the formation of H2O, then there is a decrease in the right hand side components, and according to the Le-Chatelier's principle, that tells us that when one side of the reaction is less, it is limited, then what happens? The reaction gets pushed towards that side and more of those products get formed, and therefore the forward reaction you see here, it gets increased. So more of H plus gets formed and more of CH3CO minus gets formed. Similarly, when H plus is added to this to this buffer, so to this salt, okay, what happens? More of H plus is added, so this H plus actually combines with this conjugate base and it forms this reactant. So since the reactant is more, what happens? It should actually go towards the left hand side, but then this is a salt. So since this is a salt, what we do is this only now there is complete dissociation. Since this is a salt, there is complete dissociation of Na plus and CH3 COO minus. So this is a buffer system. All right. So now buffers are aqueous systems that tend to resist changes in pH when small amounts of acids and bases are added to them. They keep the pH constant by taking up protons which are released during reactions or by releasing protons when they are consumed by reactions. So buffers are a vital components for modeling biological systems and have many uses in cell culture, molecular biology, nucleic acid, protein purification, etc. So a buffer, an acidic buffer system consists of a weak acid. This is a weak acid and the conjugate base. So this weak acid is the proton donor and the conjugate base is the proton acceptor. Alright. So now what is buffer capacity? Buffer capacity is a kind of resistance that the buffer puts up with the system, that it is not going to change its pH. Okay. So it is a quantitative measure of the resistance of the pH change when H plus and OH minus ions are added in a buffer solution. So buffer capacity depends on the number of moles of acids and bases, and it also depends on the concentration of the solution, which is after all the number of moles of acids and bases. Alright. So now this Henderson-Hasselbalch equation for an acidic buffer, it is now this Henderson-Hasselbalch equation can also be applied for buffers, and here it is just Henderson's equation. So the difference is here instead of the conjugate base we get the salt. Alright. So now these buffers, they actually maintain the pH of our body, and I am sure you know about the buffering capacity of blood. And so whenever we have these acidic foodstuffs, what happens is the acidic components, that is H2CO3 of our blood, increases, and when we have basic food substances, then the conjugate base increases, and this is how the buffer actually finally reaches equilibrium and it maintains this pH level of 7.4 by the carbonic acid and bicarbonate ion buffering system. Alright, so what are the desirable properties of buffers? So the quality of a buffer is determined by its resistance to change in pH when strong acid or base are added, and is measured in the value of its pKa, where the larger value of pKa, the weaker the acid. Alright. So the important characteristics of buffers are it should be water soluble and it should not be reacting with the components that are added into it. It should be non-toxic. Yes. 1 minute. So we were looking into the desirable properties of buffers, and amongst them we have all of these different important characteristics that you can have a look at. Now finally, the questions on the desirable properties of buffers are there. You can try to answer them. So the first question is, buffers used for biological, one minute. So questions on the desirable properties of buffers. Buffers used for biological experiments should have the following characteristics. First one is chemically stable and water soluble, which is definitely true. A buffer should be chemically stable and water soluble. The next one, it should not interact with biological molecules. That is also true. It should not. So the next one is can change pH easily. I don't think this is true, because the main purpose of using buffers is that the pH should not be easily changed and they should have high ionic strength. So let us look at the options. 1 and 2 are correct in option A. Alright. So yes. So 1 and 2 are correct in option A. Let us see what are the right answers. Yes. So the right answer is number A. And then buffers with high buffering capacity means that the buffer has a high pH value, the buffer has a high pKa value, the higher concentration of salt is present in the buffer, and the last option is higher concentration of acid or base is required to change the pH of the buffer. So the correct answer is definitely D, right? So buffering capacity is the ability of a buffer to retain its pH when strong acids or bases are added in the medium. Alright. So in other words, the buffer capacity corresponds to the amount of H plus or OH minus ions that can be neutralized by the buffer. So some very commonly used buffers have been listed here. Here you can go through the pH range of the buffers and some examples are given here. So this Beas, MOPS and Hippies buffer, these are all examples of Booth's buffers. So Booth's buffers are zwitterionic amino acid derivatives and they are very widely used biological buffers. Tris is also a zwitterionic buffer, and so all of these buffers are very common in biology labs, and also phosphate buffer is very common, tris buffer is very common. Now if you look at the buffer range of different kinds of buffers, you see that phosphate has the largest buffering range. Alright, and the next one is what? Citrate, it has a very good buffering range. Then tris is having not very low, but then it is having quite good buffering range. Hippies buffer is there. So all of these different buffers are there with different buffering range. You can go through them. So the questions regarding buffers, let us look into them. So questions on the commonly used buffers. So which of the following is or are Booth's buffers? So Hippies, MOPS, Beas, phosphate. These are the four options, and the questions are whether 1, 2 and 3 are correct, only 4 is correct, 2 and 4 are correct, 1, 2 and 4 are correct. So 1, 2 and 3. I think all of these 4 are correct. And let us look at the next question. Which of the following statements? So let us look at the answer. And the next question we see which of the following statements is or are true. So MOPS and Beas are buffers. These are examples of Booth's buffers. Tris is a zwitterionic buffer. Phosphate buffer may inhibit an enzyme's activity. Tris buffer closely resembles the intracellular environment. So which one of these do you think is true? Alright, so we will look at the answer now. So MOPS and MES buffers are goods buffers. So that is definitely true. So we look at these two now. So out of B and D, one is possibly correct. Tris is a zwitterionic buffer, which is again true. So B is likely going to be correct. And then phosphate buffer may inhibit an enzyme's activity. So phosphate buffer actually very closely resembles the internal pH environment of a cell. And so in the cell we know that at the same time all the enzymes will not be active. Every enzyme has its own optimal activity, and therefore even in the cytoplasm there are chambers. Inside the Golgi it's a different temperature, inside ER it's a different temperature, inside the nucleus, different pH conditions, inside the nucleus maybe the pH is slightly different. So that is how there are compartmentalizations inside the cell, and inside each component these respective enzymes are active at their respective pH. And so therefore the option B is likely to be correct. And if you look at this, this is the structure of pH. So yes, it is likely to be a zwitterionic buffer because all of these H plus, all of this OH minus, they get negative charge, and this NH2 it gets a positive charge, and so zwitterionic form is formed, right? So phosphate buffer very closely resembles intracellular environment, and the right answer is B. So in the next question, we see what would be the optimum pH for Hippe's buffer of pH 7.6. So where does 7.6 lie amongst all of these? So this can be a correct answer, and this can also be correct answer because it can lie here. This should not be, and this also should not be the correct answer. Alright, but then Hippe's buffer does not have this big pH range. Its pH range is rather this one, the first one, and therefore option A is correct. Now in the next question we will be looking at more questions on buffers. So buffers are made up of weak acids and the conjugate base of another acid, which is not true because buffers are made up of the conjugate base of the same acid, right? So one is definitely wrong. So if one is wrong, then all of these are incorrect. So if pKb of a base increases, then it becomes a weaker base. Is that so? For an acid, if temperature increases, the pH will get decreased. That is true. So 3 is definitely correct. Okay, and for a 10 ml buffer solution, if we add 5 ml of distilled water, then pH of the buffer gets changed. Now pH is not likely to be changed if pure water has been added to a buffer solution. So let us find out what is the correct answer. So correct answer is D because yes, so pKb is after all just the opposite of pKa, and therefore when the pKa increases it is more basic, right? So when pKb increases it should be more acidic, and therefore it becomes a weaker base, right? So buffers are made up of weak acid and their conjugate base, so of the same weak acid, right? So pKb is equal to negative logarithm of Kb. Lower Kb indicates higher pKb. 1 minute. I think someone is trying to join. Okay. Okay. So we'll continue. Buffers are made up of weak acids and their conjugate base. Alright. So for a base, as Kb decreases, the dissociation decreases and therefore the base becomes weaker. For an acid, yes, if temperature increases, the dissociation of the acid increases and therefore it becomes more acidic. Alright, more H plus ions are given out in solution. So pH of the buffer solution does not change our dilution, however the equilibrium may shift. So we have two questions on pH that are a little more thought-provoking compared to the previous questions, which were very direct. So let us look at these. So a buffer is prepared for from 65 ml of 0.2 molar 0.2 molar NH4OH and 35 ml of 0.2 molar NH4Cl. Now what is the pH of the buffer solution considering Kb is equal to this value for NH4OH? I give you 3 minutes to solve this question and then we can discuss. Okay, so let us look at the solution to this question. So when both of these buffers are mixed, the total volume is 100 ml. Alright. So in that 100 ml, if we look at the concentration of this ammonia that is formed when NH4OH dissolves in water, so the concentration of ammonia here is 0.2 into 65 by 100. So that is 0.13 molar. If you look at the concentration of this NH4 plus when it breaks up into NH4 plus and Cl minus, the concentration of that is 0.2 into 35 by 100, that is 0.07 molar in the final solution. Alright. So what happens? Ammonia combines with water and this is a forward reaction, backward reaction, and we get NH4 plus and OH minus. So initial concentration of ammonia was 0.13 when, and then after that NH4Cl was added, and so initial composition of this is 0.07 and initial composition of OH minus is 0. But then at equilibrium what happens? This changes, everything changes by X amount, and X is after all the number of moles or the mole fraction. So number of moles that gets changed. So what happens? This is getting decreased. These two are getting increased by X amount, and therefore we calculate this X from the Kb value that is already given. So Kb is equal to X into this value, right? So Kb is after all this value multiplied with X and divided by this initial value. So from there we derive X. So X comes out to be 3.34 into 10 to the power minus 5. This value is nothing but the concentration of OH minus ion, and so from there we can find out the POH. They have asked you to find pH. So once we find out POH, we just subtract it from 14 and we get pH as 9.52. Alright. So another question on pH. A researcher accidentally puts 1.6 6 grams of NaOH pellet in 1 liter of 0.5 molar acetate buffer of pH 4.74. Now what would be the change in pH? So Ka is equal to 1.8 into 10 to the power minus 5 for acetic acid, which is denoted as HAC. Okay. So I give you 3 minutes to look into this question and then we can discuss it. So let us discuss the answer to this question. So 40 grams of NaOH has been added. So 1.6 gram of NaOH pellet is added in 1 liter. So from there, now 40 gram is the molecular weight of NaOH. Alright, so 1.6 gram has been added. So exactly what is the, how many moles of NaOH has been added? It is 1.6 divided by 40, so that is 0.04 mole. Now, next what they do is, they find out the concentration of NaOH in solution, and so that is nothing but, so it is 1 liter, and so that is nothing but 0.04 molar. Now this is a very strong base. So what happens? Acetic acid and OH minus, because NaOH dissociates into OH minus and Na plus, right? So this dissociates, and so this OH minus concentration, initially you can see that it is 0, and the acetate concentration, initially it is 0.5 molar. So what we see is acetic acid, upon addition of OH minus in the form of NaOH, this actually decreases, and here this actually increases. So the new concentration of HAC and AC minus are 0.46 and 0.54 respectively. Now what happens? Acetic acid breaks into acetate ion, which is the conjugate base, and H plus ion. So these are the two new concentrations. Now these are considered as initial concentration, that is 0.46 and 0.54. Then what happens? There is change of X amount decreases from here, and X amount gets added to the conjugate base at the H plus ion, and finally at the equilibrium concentration this is actually decreased, whereas this is increased by X amount, and H plus is also increased. So what we do is we find out the X similarly as we did in the previous question. So for that what we do is we take this and we multiply it with this plus X, which is the concentration of H plus, and then we divide that with 0.46 minus X. So solving for X, we get X is equal to 1.53 into 10 to the power minus 5. So pH is equal to minus log of this value, so that is 4.82. Now change in pH is 4.82 minus 4.47, that is 0.06. Here they had already given you the initial pH, and so the new pH just needs to be subtracted from it, and so the change in pH is after all 0.08. Now we go to the last question, which is a miscellaneous question. Now they have asked you which of the following is true. So tap water is used in biological experiment. Is that true? No, that is not true. Why? Because tap water contains a lot of microbes, a lot of minerals, and we do not want all of that to interfere with the biological experiments. Alright, so tap water is definitely not used in biological experiments. Now, a thermal shaker can control temperature and rotation speed. Look at this thermal shaker. This is a thermal shaker, and if you look at the settings, this is the speed of the thermal shaker 150, and this 39.6, this is the temperature. So that means yes, thermal shaker can control temperature and rotation speed. That is true. And in thermal shaker, what happens is these are liquid media. So this liquid media is actually given into this thermal shaker, and then it is incubated overnight so that the growth of the bacteria can be carried on, and then we can get a larger population of bacteria the next morning. That is, the OD of the solution gradually increases. So this is basically done to grow bacteria in a liquid media. So that is the shaker incubator. And then for centrifuge, so 100% ethanol is used for surface sterilization. We studied in the week 0 assignment that that is not the case. So thermal shaker and cooling centrifuge are same equipment. You see that they are clearly two different equipments. So this is definitely not true. So which one is the correct answer? Let's find out. One minute. So this is rather the correct answer. Thermal shaker can control temperature and rotation speed, and the shaker incubator, in this the samples are usually spun at a speed of 120 to 200 rpm, and in a centrifuge they can be spun up to a speed of around 12,000 rpm. So in centrifuge we actually distinguish we can actually separate out components in a liquid based on their weight. All right, so now these are the references. You can follow my YouTube channel, which is into my bioverse, and thank you for your cooperation. We will be ending it here.